Hello, everyone. This is Chinmaya from the Addictive Brain, and welcome to our Interview with Scientists series. Today, we have Dr. Catharine Mielnik, who is a postdoctoral fellow at the University of Toronto in the Department of Pharmacology and Toxicology, with the research group of Dr. Ruth Ross. Her postdoctoral work has focused on preclinical validation of cannabinoid-targeting pharmacological compounds in models of neuropsychiatric disorders. Her paper titled Consequences of NMDA Receptor Deficiency Can Be Rescued in the Adult Brain was recently published in Molecular Psychiatry. In this paper, the authors address questions regarding the possibility of changes in the NMDA receptor in adulthood and the best time to treat neurodevelopmental disorders. So first of all, congratulations, Catherine, on publishing this paper.
Thanks.
How are you feeling about it?
It's 10 years in the making, right? So It kind of feels surreal that it's out there.
That's good.
This thing that I talk about, when everyone asks, what are you doing in your life? And you're like, I'm working on this, but nobody can read it. And now everybody can read it. So it's exciting.
So tell us something about the paper. What are the main findings?
So we set out to look at neurodevelopmental disorders and kind of understanding when is a good time to treat these neurodevelopmental disorders. And based on work that my PhD supervisor had done and generating a mouse model with a knockdown of a component of the NMDA receptor, we were able to use this model and kind of address that question of, do you need to rescue very early, or is the brain still kind of malleable enough that you can rescue it later into adulthood? And this really stemmed from the fact that a lot of neurodevelopmental disorders such as autism, schizophrenia, as well as another number of other pathologies, diagnosis comes late, or diagnosis isn't right when the developmental components are happening and the rewiring is happening. So there was a lot of kind of narrative in the scientific field that these neurodevelopmental disorders, diagnosis was coming too late to be able to treat them accurately. But we were able to kind of show that the brain, even into adulthood, has still a lot of capability of rewiring and reforming these connections to be able to have a rescue of, for example, cognitive deficits that are known to be very treatable. So it was exciting.
Oh, that really sounds very exciting. I went briefly through over the paper, and I found a lot of terminologies, like maybe you can tell us what are NMDA receptors?
So NMDA receptors are ion channels. They're made up of 4 different components. And for us, we look at targeting one of those subunits, which is the GLUN1 subunit, which is coded by the GRIN1 gene. So if you see it a lot in the paper, that's what the GRIN1 knockdown is that you have less of this subunit, and this subunit is essential for the proper formation of these channels. And these channels allow for the influx of ions into the neuron, which then establish these connections, which allow for these behaviors to take place, cognition, all those different types of behaviors.
That's great. And where are these NMDA receptors found in the brain?
Everywhere. So they are pretty much expressed in a lot of neurons. That's why it makes it hard to target this receptor. But yes, we did a global knockdown and we did a global rescue to the best of our abilities. Yeah.
That's great. And what is GRIN1?
So GRIN that codes for this one subunit of GLUN1, because within the NMDA receptor, you have GLUN1 subunits and you have GLUN2 subunits and even GLUN3. And then on top of that, GLUN2 can have A, B, C, D. So there's all these different players, but within every NMDA receptor, there is at least 2 GLUN1 subunits. So by targeting GLUN1, which is the protein that is from GRIN1, the gene, we're able to target every NMDA receptor.
Okay.
Yes.
And and what neurodevelopmental disorders are affected by this mutation in GRIN1?
So you have to understand we started this in 2010. So
Okay.
when we first were kind of looking at the scope of this work, we were interested in schizophrenia, autism, intellectual delay, which is just an umbrella term for cognitive deficits. So all of those neurodevelopmental disorders do have or can have components of GRIN1
Okay.
changes, mutations, implications, those types of things. But
Yes.
later on into the work, we were contacted by a group of parents who have children that have mutations in the GRIN1 gene.
Yeah.
And now with whole exome sequencing kind of taking off and being more accessible, there's
Yes.
more and more people that are being diagnosed with these GRIN1 mutations. And it's now becoming a little bit more of an umbrella disorder that has a lot of different manifestations. So some children will have certain manifestations,
Yes.
seizures, motor disorders, speech, cognitive delay, and others might have very mild versions of it. So it's just kind of all under one umbrella kind of
Okay.
term. Yes.
And what kind of mouse model was used in this study?
So the mouse model that is used in this particular paper and this study is a novel generated mouse model, but it was based on the mouse model that was generated by my PhD supervisor, Dr. Amy Ramsey,
Yeah.
from her PhD work. So basically
Okay.
what it is that the mouse model has a insertion mutation, so a mutation put into this GRIN1 gene in a
Yeah.
specific place.
Mhm.
And that leads to less expression, not no expression, because if you have no expression, it's not a knockout. If you have no expression, it's lethal.
Yeah.
So it has less. And it's all dependent on different brain regions, have different levels, and there's less GRIN1, which means less functioning NMDA receptors.
Okay.
So what we did for my PhD was we wanted to be able to reverse that mutation. And to do that, we had to generate a novel mouse model with the same mutation, but this time with the ability to cut it out. And to do that, it's a Cre-lock system that's used in the scientific community.
Yes.
And basically, these lock sites will kind of bookend the mutation.
Yes.
And these almost act as these like markers for an enzyme called 𝘂𝐚𝐿, which once it enters the nucleus, it allows it to identify these markers and then cut out whatever is in between these markers. And that was our method of reversing the genetic mutation. But is that translatable to humans? No, because we
No.
wouldn't have this type of system in a human. And it was much later into my PhD that something called CRISPR came up,
Yes.
this technique
Yes.
that would have saved a lot of time, but it's okay. That's how science is.
Yes.
Yeah, in humans, it's not a translatable system. It's really a toolkit that scientists have to manipulate genes.
Yes.
For the recombination, and that's what we use for our mouse model.
Sorry, could you repeat that? There was a lag.
Oh, I'm sorry.
No,
I'm hoping that the internet is still good here. I just basically said that we use 𝘂𝐚𝐿 as a tool for our reversal of the genetic mutation.
Okay. And you did a lot of mouse behavior in this paper.
Yes.
But as you said, these are not translatable to human patients. But
no.
But if you want to do some behavior in humans, what kind of behavior would you do?
So I just want to preface that a lot of the times, you can't directly translate mouse behavior to human behavior because, for example, anxiety in rodents is tested a lot based on like avoidance behavior. So rodents don't like to be in brightly lit open spaces. Okay. So you're trying to kind of understand the implication of their exploration of relaying it back to neurons and circuits that are related to anxiety behaviors that are seen in humans. So it's not directly relatable, as in like we don't make humans run mazes, for example,
Yes.
right? That's not something that we do. But the same circuits, the same neurons are implicated in anxiety behavior that we see in these mazes with rodents, and that's how we can implicate it. But there is one clinically translatable behavior that we have in our study, which is the pre-pulse inhibition. And you can do that same experiment or same behavior in humans, because pre-pulse inhibition is sensor motor gating, which means that it's the way that the brain filters kind of input of sensory input, basically. So your brain is getting a lot of input all the time, and it has to filter out what is important and what isn't important. And that's what you're doing in sensor motor gating. You are filtering out your input, your sensory inputs, so that you don't react to everything. You know, you don't react to everything that moves. You don't react to every sound. You don't react to every change in light or something like that. But people who have some of these disorders, that filtering system is compromised, and they are unable to kind of filter it out and are unable to control the reaction to those things. So that's what we do. So in the mouse, it is a sound that they get and they startle. So they kind of jump, and we can measure that jump and kind of the force. And if you give a much lower sound, maybe like 100 milliseconds before the loud sound, the brain then filters out that there's this sound, and then they shouldn't be as startled anymore. So you have less of a jump, right? But if you have a deficit in this kind of gating, then they will still startle even if you have this small sound beforehand, and they'll startle maybe even bigger. So in humans, you can do that with a startle response in the face, and they look at the startle response in the face, and they do acoustic, or you can also have, I think you can have response to light maybe sometimes, or I know in rodents, they can also do whisker response. So if you like puff a little bit of air onto their whiskers, they can also kind of startle to that stimulus. So it's any kind of sensory stimulus that you might have. So not all rodent stuff is translatable, but some of it is. I just want to put it out there, you know, fighting the good fight for the rodent research.
That was really expensive, but thank you for clarifying that.
Sorry.
No, that was really good. So my last question is, you injected tamoxifen. So what is tamoxifen, and why was it injected, and what does it do?
So it might sound weird that we're giving an anti-cancer drug to mice to rescue neurodevelopmental disorders. And this is something that came up when I was talking to a lot of the parents that have the GRID1 mutation children. The drug is used as a tool, and I have to just put it like it's another tool in the toolkit of scientists to be able to kind of use these gene modifying techniques. And basically what I had meant, we use tamoxifen to activate our 𝐕 recombinase. So I mentioned earlier that we use the 𝐕 recombinase lock system, and you don't want this enzyme to go in and cut out your mutation from the very beginning, because you want to be able to control it. So you use a mutated version, or it has another domain that's bound to it, and that makes it sit outside of the nucleus. So it gets sequestered in the cytoplasm. And it's not until you give tamoxifen, which binds to this estrogen receptor domain that's attached to this 𝐕 recombinase, and then it allows the 𝐕 recombinase to then go into the nucleus, and then it'll find these lock sites, and then it'll be able to cut them out. So again, tamoxifen is not used to treat neurodevelopmental disorders, but it is used as a tool. There's other systems in science, doxycycline ones, that are activated with another drug. We controlled, because tamoxifen is a really kind of nasty drug as well, because it activates, or it interacts with estrogen receptors throughout the entire body. We gave all our mice tamoxifen. So we controlled for drug effects. That's something that's important, because there's been some papers that have come out that have shown that there might be drug effects that are lasting that might kind of complicate your results.
Those were my questions. So if the audience have any questions, please feel free to write in the comment box, and we will ask Catherine about these questions. So
I
what are
know.
your next steps?
Oh, my next steps. I mean, now I've been in a postdoc for 3 years with Dr. Ruth Ross, and I kind of pivoted and did cannabinoid research.
Okay.
So we still use some of these models because some of these models are good models for psychosis or neuropsychiatric diseases. So we can still use these models without rescuing in them and just having the mutation. You can test different drugs to see if they have any kind of antipsychotic effects. So that's pretty much been my PhD. I mean, my postdoc is working on preclinical validation of cannabinoid targeting compounds. That's pretty much what I've been doing, and I kind of pivoted a little bit, but it's still all related, right?
Yes. So we have a comment from Sohini. That's a great point you just made about being careful about drug effects. We have brainstormed about using tamoxifen for a CPP study in our lab.
So I guess the comment is more that you can use tamoxifen. I mean, if you want to control and give tamoxifen to all your mice, then you have a control for drug effects. But if that's not reasonable, there are, washout periods. There's been some studies done to look at what are the washout periods for tamoxifen.
Yes.
I think within a 4-week time period, you should not have any more cognitive effects.
Yes.
So there are ways around it if you need to kind of use tamoxifen in your studies. I think just being cautious of washout periods and controlling for drug effects, that's always the best way.
Yes. There were really good comments for your PPI explanation. Everyone found it very useful. Some people understood finally what is PPI. Right,
because it's this abstract term of, that the mouse jumps, there's a sound. What does it actually mean? But yeah.
We have another question from Mila.phd. When possible, how will gene therapy look like in people? Thanks for the great talk.
That's a loaded question because we don't know. And, for example, in our mice, we were very nervous about rescuing these receptors because we thought it would cause seizures. Because if you give any kind of drug that increases glutamate signaling or NMDA receptor signaling, it tends to lead to seizures. So we were very worried that this brain had developed in hypofunction or like a decreased glutamate signaling type of environment, and all of a sudden, we're gonna bombard the system with glutamate and it won't know what to do with it. So gene therapy, I think, is a very loaded topic and a very difficult question to ask, especially in the brain. How effective is it going to be? How do you target specific brain regions? How do you do all those very specific rescues? I think it's a possibility, but it's a super cautious possibility. And I think, a lot of the times, if you have these neurodevelopmental disorders, not everybody knows what the genetic mutation is that causes them, right? Mm-hmm. You don't know. No. So if you don't know what the cause is, you can't use gene therapy, right? Yes. So then you leave a lot of people that are unable to use gene therapy. Yes. But even if you do know what the mutation is, can you rescue it everywhere? Can you not? Do you get good permeation of these rescues? Yeah. It's a hard question to answer. I'm sorry, I don't have a good answer for it.
And we have another question from Yalin Sun. Have you observed any sex differences in the GRIN1 knockdown mice?
So there's a great paper by my old lab colleague from 2014 that looked at all the different sexual dimorphisms that exist in these mice. And overall, there is no difference in later adulthood, but you can see that there are differences when you're going through adolescence, right? In general, female mice will develop sooner than males. Okay. So you have a difference in this kind of transitory period. Yes. In our mice, we saw no real differences in sex when we looked at all the different behaviors, except for one, which was a cognition test, yes, which was called Puzzle Box in our paper. And the puzzle box is basically this short-term 3-day kind of experiment where you're exposing the mouse to a problem-solving kind of environment. So they're put into one area of the box and they have to get to another area of the box. But the box is separated by a divider and it's connected by a tunnel. So you are taking advantage of the mouse's ability to, or the mouse's preference to leave this brightly lit open box and go to this dark chamber, but they have to get through a series of tasks, right?
Yes.
So the first one we start with is just an open door and an open tunnel, and they'll go through. And then you close the door and you see how long it takes them to problem solve. And then you'll fill the tunnel with bedding, so they have to dig through. And then you'll change it up and then put a plug into the tunnel. So now they have to do a different task. So you're kind of looking at different ways of changing the problem. You're looking at short-term and long-term memory. If we show it to you again after a short period of time, are you able to do it faster? Are you able to do it better? So we did that. And basically what we saw is that females in general completed the task faster than males. And we saw that the rescue was better in females. So although we were able to rescue it in both males and females, it was a little bit better in females. So I don't know what the implication of that specific behavior will be for cognition, but there were those sex differences. And just an important thing to note about sex differences in these kind of ages, we saw that if you looked at an adolescent rescue, so if you started to kind of manipulate the system during their adolescence, it was almost, you were able to get rescue, but it was much more variable. So again, when we were talking about this adolescent period, males, females are differently going through that adolescent period. So it was much more variable. So maybe that was something that was also interesting to see.
Okay. Any comments about your paper, or you want to say something?
I mean, I want to thank a lot of the people that have been along the way with me. I mean, 𝐂𝐚𝐭𝐿, you also were there for part of the journey. You know, we did have a lot of collaborations with a lot of really good scientists. So I'm very thankful for the people that were part of this journey and that were part of this whole story, kind of seeing the light and, you know, being able to address these questions that are, you know, very fundamental in terms of, you know, clinical relevance, but not necessarily immediately translatable, but it does change and shift the narrative of what the clinical implications might be for neurodevelopmental disorders. So I'm very thankful for where we are today.
Yeah, but indeed a great piece of paper. I enjoyed briefly reading it. I haven't read it completely, but I enjoyed briefly reading it.
I'm pretty sure you've heard me talk about it.
Yes.
Ad nauseum. I'm pretty sure you can point out, like, if you read it, you'd be like, I remember when she told me about this study. I remember when she told me about this. So no worries.
Thank you. Thank you everyone for participating in the question and answer session. And thank you, Catherine, for taking time out on a Sunday to talk.
Yes, no, thank you for having me. I really appreciate it.
Our pleasure. And this video will be available on YouTube in a short time.
Wow, okay.
So you will be able to share the link.
Okay, it'll be on the internet forever.
Yes. Thank you very much, and I wish you a nice Sunday.
Yes, you too. Thank you, and thank you for everyone that came. Bye.
Bye-bye.